Hello, my name is Yury Bukhman. I'm a computational biologist at Morgridge Institute for Research. I'll be talking about mammalian genome projects done in our lab. We have completed genome projects of three interesting species of mammals. Blue whale is the world's largest animal ever, larger even than all known dinosaurs. Etruscan shrew is one of the world's smallest mammals. Now rat is a close relative of laboratory mice and rats. However, in contrast to those species, now rat is diurnal. It is also an interesting animal model of diabetes. Like humans, it can develop diabetes spontaneously when fed their own diet. We have worked with the Vertebrate Genomes Project, an international consortium of researchers whose goal is to generate reference quality genome assemblies of all vertebrate species. The VGP includes some of the world's top experts in genome assembly and curation. Our genome assemblies are based on PacBio long-read technologies. First, PacBio reads are assembled into contigs. This draft assembly is purged of spurious duplications that are occasionally generated by assembly algorithms. This is followed by 3 scaffolding steps, which join the contigs into scaffolds that have some gaps but often approach whole chromosomes in length. These steps utilize 10x linked short reads, optical maps, and Hi-C. Short reads are then mapped back to the assembly and used to fix base level errors and small indels. Finally, the assembly is manually curated to fix scaffolding errors. In NowRat, we were also able to sequence the parents of the animal and use trio-binning technology to fully phase the 2 parental haplotypes. I will now say a few words about the blue whale genome. Our blue whale assembly is one of the best quality cetacean genome assemblies available. Panel A shows assembly contiguity metrics of cetacean genome assemblies in the NCBI database. Blue whale's Latin name is Balaenoptera musculus, and it is shown in red. The other 2 VGP assemblies of cetaceans are shown in blue. The rest of the cetacean assemblies are shown in black. They are all short read-based assemblies. The positions of the assemblies along the horizontal axis signify that, on average, the contigs of the VGP assemblies are almost 2 orders of magnitude longer than those of the short read assemblies. Panel B shows BASCO scores, which measure assembly completeness. By this measure, our blue whale assembly is the second place. Panel C shows the status of more than 1,800 ancestral mammalian genes mapped to the cetacean genomes by the TOG algorithm. Blue bars are intact ORFs. Blue whale is also in the second place. Mark Chiasson annotated segmental duplications in our blue whale genome. These are duplicated segments of at least 1 kilobase in length that are at least 90% identical to each other. They are not transposable elements. Rather, they are thought to arise through genome duplication and recombination errors. They often contain genes and are an important driver of evolution. They are detected by aligning the genome to itself to find near-identical segments. However, some duplications cannot yet be resolved by our assembly algorithms, so that multiple similar segments existing in the genome get collapsed into a single segment in the assembly. Such collapsed duplications can still be detected as peaks in coverage depth when sequencing reads are mapped back to the assembly. Here are some examples of duplicated genes. The genes in panels A and B are encompassed by collapsed duplications. Panel C shows a gene where some copies have been resolved and some not, as is evident from a peak in read coverage. Panel D shows a resolved tandem duplication. This gene exists in 2 copies in the blue whale, shown in blue, and only 1 copy in Vaquita, shown in orange. We compared blue whale segmental duplications to Vaquita and cattle. Vaquita is the world's smallest cetacean, while cattle is a land relative of cetaceans belonging to the same mammalian order. This plot shows the number of copies of duplicated genes versus percent identity, with collapsed duplications shown in lighter shades. You can observe that the blue whale has numerous copies of genes in collapsed duplications. Multiple copies of these genes are nearly identical, which made them hard to resolve. This is a sign of a large burst of duplication activity in recent evolutionary past. Phil Morin and Amy Lang used the genome to derive some insights into the blue whale's population genetics. Blue whales live throughout the Earth's oceans with the exception of some inland seas and the high Arctic. However, not all their local populations are in regular contact with each other, and several subspecies have been proposed. The individual that we have sequenced comes from the Northern Pacific. Another group previously published short-read sequencing data from a North Atlantic blue whale. Phil and Amy mapped the reads from both individuals back to our genome assembly. Pacific and Atlantic blue whales both show high levels of heterozygosity, evenly distributed across the genome. In fact, blue whale is one of the most heterozygous species among the mammals. This is despite having been hunted almost to extinction in the first half of the 20th century. Blue whale is currently an endangered species. However, it has been internationally protected since the 1950s and its populations are recovering. A PSMC analysis indicates that the North Pacific and North Atlantic blue whale populations became genetically isolated around the time of the last interglacial period. Now on to Nile rat. Our Nile rat colony is managed by Huishi To at the University of California, Santa Barbara. As I have said before, Nile rat is diurnal and susceptible to diet-induced diabetes like humans. It is also an important agricultural pest in Africa. Our Nile rat genome assembly is among the most contiguous and complete of the publicly available rodent assemblies. It has also been fully phased into 2 parental haplotypes. Chintao Yang has cataloged various types of heterozygosity throughout the genome, such as single nucleotide variants, large inversions and translocations, and copy number variants. Similarly to the blue whale and its relatives, we annotated segmental duplications in Nile rat, Norway rat, house mouse, and white-footed mouse. The latter is an outgroup more closely related to hamsters than to mice and rats. We have detected recent increases in segmental duplication activity in all of these species. However, the strongest burst of such activity happened in the house mouse, which had been previously reported by others. One cluster of duplicated genes we paid particular attention to was amylases. Amylase is an enzyme that helps digest starch. It had been previously reported that various species acquired additional copies of amylase when they adapted to starchy diets, including humans, dogs, mice, and rats. The Nile rat eats a low-starch diet of plant shoots and leaves. Not surprisingly, it has 3 functional amylase genes, while Norway rat has 5 and house mouse 7. On this slide, panel A shows amylase gene clusters in house mouse and Nile rat genomes, with arcs connecting homologous regions. Panel B shows pairwise similarity of amylase genes in human, Nile rat, mouse, and Norway rat, ordered according to their genomic coordinates. The dark blue square in the middle is the cluster of 5 recently diverged amylase 2A genes in mouse, which are nearly identical to each other. On the lower left are human amylase genes, which have expanded independently of the rodents. Panel C shows a trio of amylase proteins clustered by sequence similarity, which tells a similar story. We used gene disease databases and text mining to link Nile rat genes to type 2 diabetes. This table shows the number of genes in various categories of potential interest, such as those that are different between Nile rat and house mouse or affected by heterozygosity in Nile rat, which are also linked to diabetes. To sum up, we built reference quality genomes of the blue whale and Nile rat and did some preliminary analysis on both species. We hope that these genomes will be useful resources for future studies. I would like to express my gratitude to my principal investigators, Ron Stewart and Jamie Thompson, without whose leadership this project would not have been possible. We also had many collaborators and contractors who all contributed in important ways. I feel very privileged and grateful for the opportunity to work with all of these people. Thank you.